Today we have the topic respiratory system, and it is better to start its discussion directly from its embryonic development, because almost all organs of the respiratory system, they derive from the esophagus, from the hindgut, from the endoderm. And as you can see, at the end of the 4th week, so-called lung diverticulum is formed in the esophagus. And by future growth, it gives rise to the trachea and bronchi tree up to alveoli. Why is it important to understand? Because sometimes, due to improper separation of the trachea and esophagus, some abnormalities might be observed in newborn children. Those abnormalities are shown in the lower part of the slide, as you can see. And sometimes esophagus, its proximal portion appears blind-ended, so it is the problem on the way of food to pass along the digestive tube. Sometimes you may see we can see the communication between the esophagus and trachea, and it causes coughing during the feeding of the newborn child. So to understand how abnormalities are formed, how it is better to correct them, you should understand the normal embryonic development. Okay, this is only in brief to start the lecture, but along the lecture we'll return to the embryonic development because it is very critical to understand when different components of respiratory system is ready to birth. Okay, in general we have to say that the respiratory system is represented by the conducting portion and respiratory portion. So conducting performs only the function of the transport of air and different gases, yeah, but respiratory portion participates in the exchange of gases. And let's first consider the elements of the conducting portion, and of course we start from the nasal cavity, where we have 3 regions. These are the vestibule, the respiratory, and olfactory regions. As for the vestibule, this is the entrance of the nasal cavity, and it is lined with the same stratified squamous epithelium as the skin, and this is the derivative of the ectoderm. Then respiratory portion, it is lined with the respiratory epithelium, which is of endodermal region. And at last, olfactory epithelium, it is present in the concha nasalis superior, in the uppermost part of our nasal cavity, and it is also of ectodermal region. It derives especially from the olfactory plicote by its invagination inside the nasal cavity. And what cells are present there? Let's pay attention, because it is really one of the most important sensory system in our organism. So the pivotal cells in this epithelium, these are olfactory receptor cells. They are bipolar neurons in the region, and as you can see, they have long cilia on the apical end. Those cilia are immotile or slightly motile, and despite they have microtubules, and you know that cilia usually are motile organelles, but not in this case. So they express receptors to different odorants, and they increase the surface area just slightly. Then axons of olfactory cells, they penetrate the cribriform plate of the ethmoid bone, so they are not collected as, for example, our optic nerve, in a single bundle of axons, but instead they perforate the cribriform plate and are in contact with the olfactory nerve, which is the first pair of the craniofacial nerves, and this is one of the most ancient systems in our brain. So those receptor cells, they are very unique in their possibilities for being regenerated, but we'll discuss a little later. So the next type of cells, these are the supporting sustentacular cells. As you say, they mechanically support olfactory cells, as it is obvious from their name, but what is especially interesting is that they produce odorant-binding proteins, and without those proteins, the smell perception is can be realized. And one of the very relevant example is the COVID-19 infection. You know that this virus, it causes anosmia. This is a very well-known early symptom, how to differentiate from the common flu. So why people lose their smell? Because not olfactory cells are affected really, but exactly those supporting cells. This is the recently discovered fact, because those supporting cells they express angiotensin converting enzyme type 2, and this enzyme is used by the virus as to enter the cell. Olfactory cells are unaffected by the virus, by the COVID virus, but supporting cells are affected. And as you can see, if supported cells are damaged, we lose our possibilities to feel different odorants, to perceive different odorants, despite olfactory cells still work. Okay. So let's move on. And also basal cells are present, of course. They are not differentiated, and they are the source of regeneration. What is interesting is that they regenerate not only supporting cells, but olfactory receptor cells as well. And this is a very rare place in our body where neurons are readily regenerated. So this is interesting. And also brush cells, they are not shown in the diagram, but they are like chemoreceptors. They have synaptic ends at their base, they have microvilli at the top, and they perceive different substances, like common sensations, the function. Then, how odorants are perceived. Actually, I recommend you to watch a video. I'll put a hyperlink in the comments below this video, because it really explains how quantum physics really can work in our nasal cavity in perception of smell. And what is especially interesting, that really smell perception is very close to the perception of sounds. But this is not the subject of our lecture, of course, and in general and in brief, you should be aware that first odorants in our nasal cavity, they bind to the odorant binding proteins, and only in combination they could interact with the proper receptors on the surface of olfactory cells. These are the G protein coupled receptors. They activate special enzyme adenylyl cyclase, and this enzyme stimulates the synthesis of the cyclic AMP, cyclic adenosine monophosphate, and this CAMP acts on sodium-calcium channels, resulting in their opening, depolarization, discharge of neurotransmitter, and the nerve impulse generation. So this is in brief how it works. Let's not stop on details and go further through pharynx into the larynx. So in the larynx, as you know from anatomy, there are some cartilaginous muscles, and there are the true vocal folds and false vocal folds. The difference is that in the false, in the ventricular folds or false vocal folds, there are no muscles. But in the vocal folds, there are skeletal muscles, which are under the voluntary control as they participate in the speech. So what's interesting in our larynx? It is lined by with the epithelium, which is of endodermal region, but in the region of vocal folds, as they are irritated by the continuous passage of air through them, this epithelium is replaced with a stratified squamous non-keratinized. And as I told you last time, such transitions from one type of epithelium into another, these are the critical sites where different abnormalities might occur. So pay attention to the sites, and always pathologists should be suspicious in this region. And notice that there are some lymphoid aggregations in the lamina propria in our respiratory system as it derives from digestive. Yeah, there are lots of similar things. So like mucus-associated lymphoid tissue in our digestive system, some amount of this lymphoid tissue also might be observed in the respiratory as well. So what is also written in this slide, that the lining of larynx derives from the endoderm, yeah, but cartilages and muscles, they originate from the mesenchyma of the fourth and sixth pharyngeal pouches, pharyngeal arches. And this information exactly for those students who want to prepare themselves for international level MCQs like IFOM or USMLE. You know the derivatives of pharyngeal arches as well as pharyngeal pouches and pharyngeal grooves are very important those derivatives and their misdevelopment results in multiple malformations. So lots of clinical tests are based on this information. So if you want to be successful in international level MCQs, you should be aware about all the derivatives of the pharyngeal region. So I advise you to open Longman's textbook and revise this material in detail. So after the trachea, let's discuss after the larynx. Let's start to discuss the trachea. Trachea, it has four general layers, similar with the digestive system again. So there is the mucosa, submucosa, fibrocartilage layer, and adventitia. So as in digestive system, we have muscularis, externum. Here, fibrocartilage layer is present because respiratory system should be always opened. It should always perform the function of air conduction. So there shouldn't be any collapse. While in digestive system instead, if there is no food inside, then it should be collapsed to cover the food particle from all the sides. Okay, so in the trachea, mucosa is lined with the respiratory epithelium. After that, there is the lamina propria. Notice that there is no muscularis mucosa in between the mucosa and submucosa, so they are not separated and there is no exact border. And in the submucosa, some mucous glands might be present, seromucous glands, and connective tissue is a little bit more dense than in the lamina propria of mucosa. Then fibrocartilage envelope. It is represented by the C-shaped cartilages. They are C-shaped because behind the trachea there is the esophagus, and when food passes through the esophagus, if trachea will be represented by the O-shaped cartilages. Those cartilages might cause the problem in food passage. Yeah. So that's why it is C-shaped. And posterior wall of trachea is represented by fibers, connective tissue fibers, and smooth muscle cells. So it is soft and it doesn't affect the food during swallowing, the food passage. Yeah. Okay, and the outermost envelope is adventitia, so it is not covered with the epithelium. It is covered with the connective tissue with its nerve fibers and blood vessels. Okay, and let's discuss in details the components of the respiratory epithelium. From the first semester, you remember that it is called also pseudostratified because nuclei of those cells are on different levels, and it resembles really the presence of several layers. It seems that there are several layers, but really all cells are in contact with the basement membrane. So there are different cell subtypes and they different their functions. Here they are listed, like ciliated, goblet basal cells, endocrine cells, all small granular cells, then brush cells and Clara cells. Last two are labeled with a blue color as they are present in the respiratory epithelium, but not in trachea, but further in bronchi tree, at the terminal portion of the bronchi tree. But we are going to discuss them right now inside the respiratory epithelium. So the first two most common cell types in the respiratory epithelium. These are ciliated cells and goblet cells. They are not considered in details in our slides, as it is obvious. Ciliated cells, they have cilia. They are highly motile, up to 25 times per second they beat, and their sweeping motion results in the movement of mucus produced by the goblet cells. So together they provide a so-called mucociliary clearance mechanism, which is critical in the defense of our airways from different foreign agents. As you know, we inhale non-sterile air. There are lots of antigens, lots of different dust particles, and it is really polluted. And to prevent the entrance of those particles up to our alveoli, to prevent its contact with the bloodstream, then mucociliary mechanism works. What is written in summary, it is summarized here, like microbes which are attached to the viscous mucus, they passively move out due to the beating of the cilia. And to understand how it works, it is better to watch a video. Let's watch this video. So here the mucociliary mechanism is shown, and you may see here different foreign agents like bacteria, viruses entering our respiratory system. By inhaling, they become attached to the viscous mucus, as it is very dense, and it is moved by the cilia, working in the lower layer of mucus, which is less dense and viscous. Here, the only one single cilia is shown, and you can see how it moves. This is a very detailed pattern, which is reconstructed based on the scanning electron microscopy and different other methods. Notice that it stimulates the movement of fluid forwards, and here all cilia are shown together. Notice that they beat in a synchronous way, and it is very important in the effectiveness of their work. In the upper part of the video, the scanning electron micrograph is shown with the real cilia. So you may compare that this reconstruction is very realistic. So now the mucus is returned to understand the real mechanism. So cilia, they work in the less viscous layer, in the lower layer of the mucus. In the upper layer, dust particles and different microbes are entrapped, and like in the escalator, they are passively moved out of our respiratory airways to the pharynx. But here the very, very slow motion of cilia are shown for us to understand the tiny mechanism, but now the real speed is shown. As I told you, up to 25 times per second each cilia beats, so very quickly, and it is invisible really. So the mechanism is very effective, and if we look at the real video microscopy, we can guess how quickly this movement is by the dust particles. Here you may see those dark points. These are the dust particles which are moved by the beaten cilia here. Okay, let's return to our slides, and altogether this is called mucociliary mechanism, and really the viscosity of mucus is a very important factor, and sometimes it is increased. So in pharmacology department you investigate lots of drugs and lots of ways how to make this mucus a little bit less dense to facilitate its expel out of the respiratory airways. Okay, except ciliated and goblet cells, also there are some small granular cells. They are called also Kulczycki cells and enteroendocrine cells. These are the homologs because they derive from the same source. If you remember, in the digestive system, some endocrine cells were dispersed among the epithelium of the intestines as well as the stomach. The same is true for respiratory system. And as it is written here, they produce different hormones. They have granules located at the base of the cell, small dense granules, as it is shown in the transmission electron micrograph here. And why it is important to know about them? Because they might give rise to tumors, and those tumors they produce hormones, and that's why some patients they suffer from different syndromes resembling endocrine pathology or increased blood pressure in case of catecholamine expression, for example. So those cells just well known not only in the normal condition but in the pathology. Then brush cells. Brush cells are homologs of tuft cells in our digestive system. They are the chemoreceptor cells, and as it is written here, they have microvilli at the top, they have synaptic nerve endings at the base, and general sensation is their function, as well as it is written, they might be also involved in absorption locally, and their function is not very well understood up to now. And next type of cells, as I told you, brush cells and Clara cells, they are not found in trachea, but in a more distal portion of our respiratory tree. And as for the Clara cells, I want to highlight that recently, in the most recent Junqueira edition, you have as a basic textbook, Clara cells have changed their name into club cells. Why? Because, as it is written, Max Clara was the Nazi supporter, and he performed his experiments on humans. And really, he examined lung samples after the intoxication, and he found that such people, they have increased number and function of Clara cells. And that's why his name now is in oblivion, and it is expelled out of histology textbooks. And that's why maybe in some MCQs you still might found the name Clara cells, but in the most modern editions the name is changed, so be attentive, because club cells were not widely used before. Then their main function to produce some components of surfactant. Also, they produce enzymes to break down mucus, and also they participate in the detoxification, as I told you. Just so remember, our lungs, they perform function not only to exchange gases, but also the excretory function is present, as some components like alcohol, it evaporates from the surface of our lungs, and such substances can be excreted not only with the kidneys, but in this way. So cytochrome works here, helping to detoxify different toxins possibly present in the air. And also they participate in the immunity by producing the secretory component of the immunoglobulin A. You know, this immunoglobulin is present everywhere in our mucosal envelopes, in salivary glands. You remember also duct system produces secretory components, and also antimicrobial enzymes. So by their appearance, they have really club-shaped, dome-shaped apex, and here in the scanning electron microscope, we can exactly distinguish them on the background of ciliated cells. Here, ciliated cells are located, and these are the club cells. Large apical portion containing many secretory granules. As you see, they produce lots of different substances. Okay, and basal cells. As for the basal cells, remember, they are the source not only of regeneration, but also of different tumors. So you should remember about them. And there are two progenitors, the proximal and distal progenitor in our respiratory system. So distal progenitor works in alveoli. So alveolar cells are regenerated independently, while in the proximal airways before alveoli, there is the proximal progenitor. It gives rise to these are the neuroendocrine and non-neuroendocrine cells. So they split early, and neuroendocrine cells, they give rise to secretory cells, which are goblet cells, club cells, and ciliated cells. What is important is that neuroendocrine cells, let's look here, yeah, neuroendocrine cells, they can give rise also to ciliated cells and goblet cells as well. So they are not terminally differentiated, despite they have granules, despite they produce some hormone-like substances causing the paracrine regulation. They still bear the possibility to divide. So small cell lung carcinoma might arise from such cells. That's why you should remember about this possibility. So neuroendocrine cells can give rise to tumors because they are not terminally differentiated. Okay, and then as for the cellular composition of our respiratory epithelium, as you can see, respiratory epithelium is not uniform, and its composition really changes along the respiratory tree. So in the trachea, some cells like goblet cells, ciliated cells, and basal cells, they are the most numerous. But in the terminal bronchial, in the terminal elements of the conducting portion, we can see appearance of the brush cells and club cells or Clara cells. Yeah. And take it in, take out. Okay. So after we have discussed the structure of trachea, and in general we have discussed the structure of the respiratory epithelium, we should also consider the structure of the bronchi tree. From anatomy, you remember that there are two lungs. In every lung there are lobes. In every lobe there are some segments. Inside the segments there are lobules, and lobules they are separated one from another by the connective tissue. So inside lobules, we can also consider some acini. So lung acinus is the morphofunctional unit of the lung. Okay, this is for the anatomical classification, but in histology, we roughly divide bronchi into three conditional categories. These are the large, middle, and small bronchi, depending on the diameter, but depending on the structure of their wall. So let's consider what are the features of all those types. So, as you remember, trachea, it has C-shaped cartilages, and the difference between the primary bronchi and trachea is only in the presence of the O-shaped cartilages. So they are continuous and they never collapse because they have O-shaped, ring-shaped cartilages. After that, large bronchi are shown, and large bronchi, they have discontinuous cartilage, so large islands of cartilage are present around the diameter, and what is present also is the muscularis mucosa. It appears in the large bronchi. As you remember, in trachea there is no muscularis mucosa, but here it appears. After that, middle diameter bronchi, the feature is that cartilage, hyaline cartilage, is replaced with elastic cartilage, and small islets of cartilage present in the fibrocartilage envelope. Then inside small bronchi, they have no cartilage, they have no glands in the submucosa, and they start to acquire brush cells and Clara cells. So here, as you can see, I gave you not the structure of every type because they have generally the same structure, but differences. So in the large bronchi, cartilage is discontinuous comparing with the primary. In the middle bronchi, elastic cartilage, elastic tissue replaces the hyaline. And after that, in small bronchi, cartilages disappear at all, as well as glands in its submucosa. Okay, so branches of small bronchi, they give rise to the so-called lung lobule. Lung lobule is sometimes considered as the morphofunctional unit, but I would like to call it a morphological unit or structural unit because it doesn't perform the function of the lung. Yeah, it is represented by different elements, but structurally it is really separated by connective tissue. As in all other organs, we call them lobules because they are separated by connective tissue. Okay, but lung lobule, it contains terminal bronchiola and many other, and their divisions. But lung acinus is exactly the morphofunctional unit, because lung acinus, it consists of different branches of terminal bronchiola, and it includes respiratory bronchioles, alveolar ducts, and alveolar sacs. Why we call acinus is functional unit? Because all elements of the lung acinus participate in the exchange of gases. Even in the respiratory bronchiola, here, we can see alveoli in their walls. So this bronchiola participates in the exchange of gases. Okay. So lung acinus, it is the territory which is supplied by one terminal bronchiola. And inside every element of lung acinus, exchange of gases takes place. While if we return back, in the lung lobule, we can find terminal bronchiola and respiratory bronchiola, alveolar sacs, and alveolar ducts. So terminal bronchiola, they do not participate in respiration. That's why we do not consider lung lobule as functional unit of the lung, because terminal bronchiola doesn't perform the function of gas exchange. Okay, let's move on. So here a schematical diagram of the lung acinus is shown. Notice that this is the respiratory bronchiola and it has alveoli in its wall. Notice that it is embraced by smooth muscle elements, and of course it doesn't contain cartilage, as well as small bronchi. And the question is, if there is no cartilage, how it maintains its lumen opened? And the answer is that here there are smooth muscle cells, and they form the backbone of those bronchioles. And besides, they are the target of many pharmacological drugs, because exactly here there is no cartilage, so those elements might be collapsed due to allergic reaction under abnormal circumstances. You know, when people inhale some innocuous antigens like the cat's fur or some dust particles, they start coughing and they feel problems with exhaling the air or inhaling the air. That's exactly because due to allergic reaction, histamine stimulates swelling and contraction of smooth muscle elements, and as there is no cartilage, they might be collapsed completely. So different drugs, relaxant smooth muscle cells, should be inhaled to solve the issue. Okay, so here I just want to emphasize how important to understand the structure of our respiratory system, how important to understand histology, to understand pharmacology in future. And if you really want to cure patients with asthma, this is the segment and this the targets for those drugs. Okay, let's move on. So lung acinus, it is represented by the respiratory bronchiola. After that, alveolar sacs, alveolar ducts. So the terminal portion of our lungs, these are alveoli, and here two types of cells are found. Actually more, but in general two. These are the type 1 alveolocyte and type 2. Also they are called pneumocytes, so they are equally used in the literature, so remember both synonyms. So pneumocytes or alveolocytes, type 1 and type 2. Type 1 cells are called respiratory cells. Okay, it is not correct. Type 2 cells are called secretory cells. And despite type 1 is less numerous, nearly 40% of all alveoli lining, nearly 40% by the number, but they line more than 90% by the surface. So type 1 cells, they are very large and they are flattened. So one cell covers many more surface area, much larger surface area than type 2 cell. Okay, never mind. So type 1 cells, they are less numerous, but they line predominant surface area of our lungs, and they participate in the exchange of gases. Type 2 produces a special liquid, which is called surfactant, and surfactant is needed to prevent alveoli from collapsing. As I told you, in trachea and bronchi tree, we have a cartilage skeleton, cartilage backbone. It prevents collapsing. In small bronchi, as well as in bronchiala, there are smooth muscle elements, and they regulate the diameter of those airways. But in our lungs, the wall is very thin to provide exchange of gases. And at the same time, those thin-walled vesicles, they always should be opened. And to maintain their lumen opened, they have surfactant lining them from inside. And this liquid is very extremely important and is produced by the type 2 cells. As for embryonic development, as I told you, we are going to return along the lecture to different aspects. Alveoli, they developed at the end of the 7th month in the number enough to provide the exchange of gases. So it is important in neonatology to understand when a respiratory system is ready for the birth. Yeah. So to survive, at the end of the 7th month, alveoli are formed. And what is especially interesting, that at the moment of birth, you know that the volume of our lungs is much smaller than in adults, and only 1/6 of all the alveoli present. And during next 10 years, alveoli are continuously developing, and alveolar sacs are formed, and even respiratory bronchiala, they appear along the next 10 years of our life. So lungs are not fully developed at the moment of birth, but they are ready to birth several months before the birth. Then lung macrophages. This is the third cell population, very important. They belong to the mononuclear phagocyte system. They derive from monocytes. So their main function, the phagocytosis of different dust particles and debris material possibly present in the terminal portion of our airways. As we have discussed, in the upper part of our respiratory system, the conducting portion, mucociliary clearance mechanism works, and its sweeping motion results in the cleaning and in removing of all those dust particles. But if the air is very polluted, if there are too much dust or too many particles, they might reach even alveoli, and their macrophages monitor and provide the surveillance. Sometimes they are called also dust cells because if they are full with dust particles, they might stay in the lung and reside here for a relatively long period of time. So let's besides discuss type 2 cells. As I told you, type 1 alveolocytes or pneumocytes, they participate in the exchange of gases, so they are flattened cells, and through their cytoplasm, gases freely diffuse. Then type 2 cells, they are large, and they produce surfactant. They have characteristic feature I want you to remember. For MCQs, it is very important. They contain so-called lamellar bodies and multivesicular bodies. Lamellar bodies, they are called so because surfactant, it is represented by the upper phase and hyperphase, and the upper phase is represented by phospholipids, like our membranes, if you remember. And layers of those phospholipids, they resemble, how to say, they not resemble, there are many layers of phospholipids inside those lamellar bodies. Lamella means like plate, and layers, those phospholipids representing the upper face of the surfactant, is the nature of those lines. That's why they were called lamellar. Okay, and multivesicular bodies, they contain components of proteins which are the hyperphase of the surfactant. So they fuse multivesicular and lamellar bodies one with another, and they add the cytostom to the surface, covering it. Yeah, and what is important that alveolocyte type 2, they can regenerate both types, type 1 and themselves. So this is a kind of the stem cell. As I told you, there are two types of stem cells, proximal and distal progenitor. Proximal works in the respiratory epithelium, and here distal progenitor works, and it regenerates both types of alveolar cells. Okay, here besides alveolocyte type 2 is shown again, so you may see lamellar bodies and multivesicular bodies, both produced in the upper face and the hypophyse of the surfactant. Here again another representation with a transmission electron microscopy of lamellar bodies and multivesicular bodies. And what is written about the surfactant? Again, as for the embryonic development, type 2 alveolar cells, they appear only at the 6 months of embryonic development. So before this term, surfactant can be produced as there are no type 2 cells. But nowadays, artificially produced surfactant is used to inject, and even children born much earlier this term, they can survive, even if they have their own alveolar type 2 cells, if the alveoli are not properly formed. Okay, so different another techniques are used to assist in breathing. Okay, and surfactant is called so because it reduces the surface tension and it prevents alveoli from collapsing. Let's move on. This is as for the components. I don't ask you to remember and to pronounce because it is really very difficult, especially if you're not native speaker. Yeah, so the upper face is represented by phospholipids, by lipids, yeah, hydrophobic in nature, and the lower face, hypophyse, is aqueous. And here it is shown, you may see like alveolar macrophages, they reside in the lower face of the surfactant, and here phospholipids are shown. Remember those molecules from the topic plasma membrane. And this is the last slide of our lecture, and we have to mention that in our lungs, air doesn't percolate through the blood. There is no direct contact between the air and the blood. And there is always a barrier between them. You know, lots of barriers in our organism. You're already aware about the blood thymus, about blood-brain, very well-known barrier, and blood-air barrier. You should know the components. Cytoplasm of endothelial cell, of course, all capillaries, and you know our lungs, they contain continuous capillaries, so the endothelial cells, they are continuous. Their cytoplasm is the first component of the barrier. Then basement membrane, as you can see, membrane is common, is fused for the endothelial cell and for alveolar cell, so the fused basal membrane. Then cytoplasm of alveolocyte type 1. Type 1 because exactly this is the respiratory epithelial cell. Besides alveolocytes, they resemble endothelial cells. They have similar functions and structure really. And at last, surfactant, which is present inside alveoli, and it represents the fourth obstacle on the way of microbes. So if any microbe present inside alveoli, it should first penetrate surfactant, cytoplasm of alveolocyte, basement membrane, and cytoplasm of endothelial cell to enter the bloodstream. So it's very difficult, yeah, and that's why infection should not be spread through the bloodstream from our lungs in case of infection, effects of pneumonia. Okay, remember that also our alveoli, they are communicated one with another with a special pores of cone. So those pores are needed to equalize the blood pressure between different alveoli. And what is even more important, if some alveoli are blocked, they can't get the air through its respiratory bronchial, so they might get some air from neighboring alveoli and still participate in gas exchange. So this is like compensatory reaction. Okay. And This is the end of our lecture. Please, if you want to get the information about recently published lectures, then subscribe to our channels and thank you for watching.